Hi there, this is Jillian Cullen on May 3rd, 2025. Today I'm going to be talking about macromolecules and how they move in and out of the cell and how they're created in the cell. I decided for my steroid hormone to choose cortisol, and steroid hormones in general are primary messengers, which means they can cross the cytoplasmic membrane because they have fat-soluble properties. Our cell membrane or animal cell membranes keep non-soluble fats out, and cortisol is a primary messenger, is a steroid hormone, which means it can move in and out of the cell freely. Here with this image you can see, I don't know if you can see my pointer, cholesterol on the top is moving in via the transport protein STAR. Cholesterol begins outside of the mitochondria and moves into the mitochondria via that transport protein, and then turns into pregnenolone, and then moves into the endoplasmic reticulum, where there are three processes that take place, three very in-depth processes that take place to turn it into cortisol. And there are enzymatic reactions in the endoplasmic reticulum that take place to turn it into cortisol. And cortisol is created and put into the mitochondria, where it is then leaving the cell to go into the bloodstream. Cortisol is a stress hormone, so it reacts when we are stressed and can help our body but can also hurt it. Next, for my protein hormone, I chose glucagon. I think glucagon's really interesting. When our body is fasting or when our glucose levels are scarce, glucagon regulates our blood glucose levels, or our blood sugar levels, which is what we see like hemoglobin A1C or just a fasting blood glucose level. Here it is moving, so it starts in the outside the cell membrane, and then there's a glucagon receptor. And these glucagon receptors, these cells are primarily found in the liver. And then glucagon is released by pancreatic cells when there is that decrease, the notable decrease in blood sugar levels. And then it binds to that glucagon receptor on the cell membrane of the liver and makes its way into the cytoplasm through that glucagon receptor. And then the decrease in glucose activates CAMP. It also activates this G protein and adenylate cyclase, and pKa as well. Both of these aid in the creation of glucose. Glucose 1-phosphate is then created into glucose in the nucleus, where that glucose is then released into the bloodstream, out of the nucleus and out of the cell into the bloodstream. This is beneficial for working with, I think glucagon can work both ways. It works in increasing our blood sugar as well as working with insulin to decrease our blood sugars when there's a notable increase, and then when there's a notable decrease, it brings it back up. For the next intracellular proteins, I think I did a generalization here because intracellular proteins do a whole lot for us. They are responsible for what goes on in the cell and how the cell is formed. They are helpful with cell organization, they remove the bad stuff of the cell, and they also maintain all of the chores around the cell. They work primarily in the plasma membrane, and they send nutrients across the plasma membrane. And they also are helpful in receiving messages and signals from outside of the cell. For example, with the glucagon, that's what receives that signal that our glucose is too low or that our glucose is too high. Sweet.